Good morning, everybody. We will continue our lecture on small DNA viruses, lecture set number 10. Last time we stopped on slide number 16, family of Polyoma viridae. So, Polyoma viridae viruses are those viruses that use animals and humans as their host, broad host range. Prime examples of viruses in this family are polyoma virus and vacuolating virus. The genome of this virus is circular, double-stranded, 5 kilobase, and the virus genome is condensed by histones inside the virion. We've briefly touched on histones before, but I have a slide that describes histones in detail for those of you who may have forgotten about the concept. Polyoma viridae family of viruses have icosahedral capsid, 3 structural proteins, and the size is 45 nanometer. Two important aspects of this family of viruses are: one, replication of the virus occurs in nucleus, and viral replication triggers and stimulates host cell DNA synthesis at the same time. Slide 17. There are some genuses in this family. The genus is Polyomavirus, SV40 simian vacuolating virus, JC and BK virus that we discussed last time. The types of infections depending on the host, we have acute or persistent infection, non-permissive or abortive infection, and if the host is newborn hamster, we see transformation or tumor production. Let's move on to slide number 20, which is the subject of today's lecture. SV40 genome double-stranded DNA. The genome is circular, supercoiled, and is condensed by histones. Polyomavirus has a 5 kilobase genome that may integrate into the host cell DNA. There are very similar, a lot of similarities between viral DNA and host cell DNA, and these similarities are the presence of ORI, or origin of replication, on viral genome, presence of promoter and enhancer. The gene expression process of SV40 genome is divided into early and late gene expression. During early gene expression, we have one early pre-messenger RNA that will be translated into 2 non-structural proteins, whereas the late gene expression, even though it still codes for 1 pre-messenger RNA, however, it codes for 4 proteins, 3 structural proteins, and 1 non-structural protein. Slide 21 is the slide that I told you before. The histones, types of histones, and functions of the histone in compacting DNA strand and regulation of chromatin configuration during gene expression. Slide number 22, SV40 genome, the control regions. SV40 genome has a region called ORI, ORI, which is an abbreviation for origin of replication for DNA. It has two regions called PEPL, P for promoter, E and L for early and late mRNA. So PE promoter for a region for early messenger RNA production, PL promoter region for late mRNA. Then we have a region called 21 base pair and 72 base pair. These are enhancer regions. So we have origin of replication, we have enhancer region, and we have promoter regions. Then we have a region called T region that it has three different binding sites for large T antigen. I'll talk a lot more about this large T antigen. T for tumor. So large T antigen is a multifunctional protein. I'll get into it. It has regulatory functions, it causes cancer, etc. Slide 23 is the process of gene expression, which I'm sure you guys have had some of this in previous courses, but for the sake of completion, I want you to familiarize yourself with this and freshen up your memories. Slide number 24, the same token, it talks about promoter region and enhancer region. What are they? What they can be found and what is their function? On the right-hand side of slide number 24, we have a four-step diagram showing how we use an enhancer, different components, and I want you to fresh up your memory on these two concepts. Slide 25, SV40 entry and uncoding. Entry of SV40 is through receptor-mediated endocytosis after it gains entry into the host cell cytoplasm in a vesicle. It will be transported to host cell nucleus, where the vesicle will fuse with nuclear membrane, and this way virus will gain entry into the host cell nucleus. And uncoding, which is separation of viral capsid and genome and release of viral DNA, occurs in host cell nucleus. Slide 26. After the viral genome is released into the host cell nucleus, we have early gene expression and late gene expression. Remember, I said that the virus is double-stranded DNA. So for early mRNA transcription, virus uses the first strand counterclockwise. Since it's circular, it goes to the left, if you wish, not to the right, counterclockwise. For early mRNA transcription, we use early promoter or PE. The DNA contains a sequence, a consensus sequence called TATABUX, that is a sequence where host cell RNA polymerase II will sit on, and the viral enhancer region is where host cell DNA binding proteins will bind. Activators or transcription factors are examples of host cell DNA binding proteins. The result is the production of one early messenger RNA. This one early messenger RNA, through the process of alternative splicing, codes for two messenger RNA that these will code for two proteins, small t antigen and large t antigen. Slide 27 describes SV40 early proteins. As I said, these are large T antigen and small T antigen. Large T antigen is a multifunctional protein, and it does a series of functions, that all these functions are to the advantage and benefit of the virus and detrimental to the host cell. See what's going on. The first thing that large T antigen does is to activate host cell cycle, meaning that it binds to and inactivates those regulators of a host cell cycle. These were P105RB and P53. Remember, through different mechanisms, these two proteins regulate cell cycle, so cell cycle can proceed in a very regulated manner. In the absence of these, cell cycle goes out of control and contributes to tumor formation. Large T antigen also blocks cell apoptosis or programmed cell death. We talked about apoptosis before. It's a genetic and tightly regulated program where if the damage to the cell is beyond repair, the cell is programmed and destined to die. So apoptosis is actually a very interesting program. We see that during immune regulation, during development, etc. And if we block apoptosis of host cell, it means that the virus has a place to set up shop and can grow and divide and replicate. Large T antigen also triggers viral DNA replication. It also serves as a negative feedback loop. Once enough early messenger RNA is being produced, resulting in the production of large T antigen, the produced large T antigen will block further early mRNA production and help the genome to switch to late mRNA expression. And also large T antigen participates in the assembly of the virus, meaning that once the genome is replicated, has made more of itself, and viral capsid proteins, the structural proteins are being made, helps them to, through specific interactions, assemble and form virions. Whereas small T antigen is not multifunctional, it allows viral DNA replication in certain host cells. Moving to slide 28. SV40 semi-conservative DNA replication. For replication of SV40 double-stranded circular DNA, we need to form something called IC, initiation complex, which is composed of viral T antigen that codes for a number of enzymes and regulatory proteins, and cellular DNA primase, DNA polymerase, and cellular DNA binding proteins. I told you these can be activators, these can be co-activators, or transcription factors. On the bottom of slide 28, you see a lot of YouTube video clip links. These are specifically for those individuals who may have forgotten about some of these processes of protein synthesis, DNA replication, leading strand versus lagging strand. I have carefully selected these videos after watching a huge number of them. So I would like you guys to look into it and freshen up your memory. Moving to slide number 29. After early mRNA production, SV40 DNA needs to be replicated to make more of itself. The process is as following. First, viral T antigen that has been produced as a result of early mRNA translation will bind to the ORI, or origin of replication of viral DNA. The viral double-stranded DNA opens up, an RNA primer is being made by host cell DNA polymerase. DNA synthesis of SV40 or DNA replication is bidirectional, meaning that we have both continuous and discontinuous strands, and the result is the production of two double-stranded DNA loops that we see on the bottom portion of the picture on the right-hand side. Moving to slide number 30, it's basically a little bit of a description of what I just mentioned to you regarding replication of SV40 DNA. Slide number 31, after DNA replication, we have late mRNA transcription. Late mRNA transcription follows viral DNA replication. It uses the second strand and reads clockwise, as opposed to early mRNA transcription that was using the first strand and counterclockwise. For late mRNA transcription, T antigen, large T antigen binds near origin of replication, or ORI, and blocks early mRNA. Remember, a few slides back, I mentioned that large T antigen serves as a negative feedback for early mRNA. It stops it. T antigen binds to enhancer and triggers late promoter, or PL, promoter for late genes. The result is production of one late messenger RNA that, through alternative splicing, makes two messenger RNA and codes for VP1 messenger RNA, VP2, and VP3 messenger RNAs. So SV40 mRNA is being transcribed by now. We are on slide 32. After mRNA production, we need to have proteins. mRNA needs to be translated into proteins. So we have two late mRNAs that both have two ORF, or open reading frames. Therefore, each mRNA codes for two proteins using two different start codons. VP1 messenger RNA codes for a protein called VP1 and agnoprotein, whereas VP2 and VP3 messenger RNA code for VP2 and VP3 proteins. Slide number 33 is just a depiction of these two processes: early gene expression and late gene expression of SV40. I strongly encourage you guys to take a look at this and make sure you get a good grasp of it. Moving to slide number 34, SV40 maturation. let's take a step back and talk and see what's going on so far. Virus comes in through receptor-mediated endocytosis. The vesicle will move or transport it to nuclear membrane, fuses to the nuclear membrane. The viral encoding and release of genome occurs in the nucleus, where we have early enhancers, early proteins that code for early genes. These early genes are large T antigen and small T antigen. Large T antigen is multifunctional, not only inhibits the function of RB and P53, it also triggers host cell DNA synthesis, cell division, which is required for expansion of virus, and production of host cell DNA polymerase. Then the large T antigen will basically serve as a negative feedback to block early mRNA. Genome is being replicated at this time by T antigen binding to origin of replication, TATA-box, promoters, and enhancer that we discussed in previous slides. Then late messenger RNA will be produced, go through alternative splicing to make agnoprotein, VP1, VP2, VP3 proteins. VPs are viral proteins. These are structural proteins. We have, at this point, we have genome, we have capsid proteins. They assemble together, and genome will be compacted by the action of histones, and the assembled virus will get out of the cell through lysis. So, assembly of virus occurs in host cell nucleus, and then virus is released from the host cell by killing the cell, or host cell lysis. One virus comes in under laboratory and cell culture conditions. One virus can produce approximately 10 to the 4th or 10 to the power 5 progeny viruses that, upon lysis of the host cell, they can go and infect other host cells. Moving to slide 35. I also mentioned that in some hosts, SV40 has a non-permissive infection or abortive infection, meaning that we have viral DNA. It cannot replicate in the host cell. The genome cannot make more of itself in the genome. What happens is viral large T antigen stimulates or triggers host cell DNA replication, resulting in host cell division and continued stimulation and division of the host cell may lead to host cell transformation or tumor production or cancer production. Sometimes viral DNA can integrate into host cell DNA through a process called the random recombination event, that in this instance we have stable cellular transformation because viral T antigen is continuously and constitutively present in the host cell. As a matter of fact, in the laboratory, when we work in the laboratory, we get some cells and we want to immortalize them and convert them from primary cells to cancer cells, so we can continuously have them in cell culture. For various studies, we transfect them with SV40 large T antigen. Moving to slide number 36 is basically a description of what I just mentioned to you guys. Moving to slide number 37. So SV40, because of its unique structure, can be used as an expression vector. So it can be used as a recombinant DNA tool in recombinant DNA technology. P that you see in front of the SV40, it says PSV40T7 expression vector. P stands for plasmid, so it's an SV40 that we can insert genes of interest and make those genes to be expressed and have more of them. So how do we do that? If we have a gene of interest, we insert it into a region of the SV40 genome called polycloning site. I'm sure those of you who have taken other courses or have worked in the laboratory are familiar with this concept. There's a region that we cut them with restriction enzymes, which are nucleases, open the two sides up and insert the gene of interest. Then we will transfect the cell with this expression vector, and because of the strong promoters that it has, it will propagate and make more of that protein. So SV40 has a very strong early promoter and has polyadenylation sites. These are the two characteristics that makes SV40 an ideal tool for recombinant DNA technology being used as an expression vector. Moving to slide number 38. Well, remember I said another virus in this family is JC virus. Let's see what are the infection and diseases caused by this virus. It can cause primary infection, which is very common in young children. The route of transmission is through respiratory route. It can also cause persistent latent infection. During latent infection, we do not see any infectious virus. We see infection in the a latent infection in the lungs, kidney, lymphoid organs. Like any other viral infection, host immune system plays a major role in curbing the infection. So if we have immunosuppression in the host, that leads to uncommon activation of the virus that can travel and spread to brain. And patients present with a very rare but fatal type of illness called progressive multifocal leukoencephalopathy, or PML. In addition to primary and persistent latent infection, virus can also take a conventional slow course of infection where we have lesions and sites of demyelination, leading to loss of memory, speech, and coordination. If you guys remember, myelin is a sheet that covers the nerve cells, and demyelination is the process of destruction and loss of those myelin sheets. This kind of JC virus infection can be seen in about 5% of AIDS patients, mainly due to immunosuppression of the individuals. Moving to slide 39, BK virus infection or disease is a common mild respiratory tract infection. It can also cause persistent latent infections of lung, kidney, and lymphoid tissues, just similar to JC. If the host is immunocompromised, then it can lead to activation of the virus and more dissemination of it. In bone marrow transplant recipients, it causes hemorrhagic cystitis, And in renal or kidney transplant recipients, it causes nephropathy, which is damage to the kidneys, and graft failure. Moving to slide number 40, the next family of viruses are the family of Papillomaviridae. Papillomavirus, these are very large virus family, more than 200 different types of viruses in this family. They are circular, double-stranded, 8 kilobase viruses. The DNA of the virus is condensed in virion by the action of histones. Papillomaviridae family of viruses are a little bit more complex than polyomavirus. We'll talk about them. These are, again, icosahedral capsid, have icosahedral capsid, 2 structural proteins as opposed to 3 that we saw in polyomavirus, and the size is slightly bigger, 55 nanometer. Similar to polyomavirus, papillomaviruses replicate in host cell nucleus, and this process also triggers host cell DNA synthesis. Moving to slide 41, the genus is called papillomavirus. It has nipple-shaped projections and causes tumor. The virus, papillomavirus, has a very restricted host range and tissue tropism. It infects epithelial cells of skin and mucous membranes. However, it's a very difficult virus to grow and culture in vitro. One of the viruses in this family, are called human papillomaviruses, HPV, 58 different serotypes of it. Usually, they cause benign tumors and warts in the oral cavity, pharyngeal area, and skin. Small number of viruses will cause a persistent infection, leading to malignant or tumor transformation of the host cell. And these viruses are a significant cause of cancers in humans. These cancers can be oral or pharyngeal, as well as cervical or penile carcinoma. Cervical, cervix, we see it in females, and penile, we see cancer or carcinoma in the penis of male hosts. Moving to slide 42, the HPV genome is double-stranded DNA. This 8 kilobase can exist as an episome in host cell. Similar to polyomavirus, it has promoter regions, enhancer regions, origin of replications, and it's also very similar because of the presence of these structures. It's very similar to host cell DNA as well. Again, we have early gene expression, late gene expression, slicing of pre-messenger RNAs, just the exact same concept that we discussed earlier. HPV serotype 16 produces some early proteins. These early proteins are called E6 that binds to host cell P53 protein, and E7 that binds to host cell P105RB protein. And these proteins are very similar to SV40 large T antigen. When you think about it, if P53 and RB are inactivated and we have something like large SV40 large T antigen, it predisposes the cell for transformation or becoming cancerous. Moving to slide number 43, there is a little bit of a description of episome for those of you who may have forgotten. Episomes are extra chromosomal genetic material that can replicate by themselves, and they can also be incorporated or integrated into the chromosome. I would recommend you guys to take a look at these definitions. Moving to slide number 44. This is the stages or steps of HPV infection, human papilloma virus infection. HPV is tropic for epithelial cells of skin and mucus, means that it has a tropism for these tissues, meaning that it infects these tissues. Transmission of HPV is by close contact. Virus infects via production of microlesions, and these microlesions enter those proliferating or mitotic basal cells as shown in the figure above. And when you think about it, virus life cycle is very closely tied to cellular differentiation, host cell differentiation. Moving to slide 45, HPV replication. HPV replication causes non-permissive infection of basement epithelium cells. It expresses early viral genes and which code for early viral proteins, and viral DNA can exist as an extra chromosomal entity called episome. So we have viral DNA there. What will happen? Viral DNA will replicate when host cell DNA replicates. So it's kind of synchronous. The two replicate together. Result is expression of late viral genes. Virus will be assembled. And we only see variants of viral particles in the uppermost layer of differentiated keratinocytes. You know, the top layer of the skin contains keratinocytes, those cells that contain keratin in them. And we see virions only in those differentiated keratinocytes in the uppermost layer of the skin. The patterns of HPV infection are asymptomatic, acute, persistent, and transformation. So the virus is capable of causing all sorts of, all patterns or ranges of infection. Moving to slide 46. Sexually transmitted diseases or STDs. Don't mix this up with standard deviation that we use in a biostatistics course. So a question that's been posed for a long period of time is why sex isn't fun anymore. The reason why sex isn't fun is because of the presence of these vast number of viruses that can easily cause devastating sexually transmitted diseases. These viruses are human papillomavirus, HPV, herpes simplex virus, human immunodeficiency virus, and hepatitis B virus. These four viruses that I just mentioned can be transmitted during sex. So that's one of the reasons that safe sex is being highly recommended. And STDs can be considered as an American epidemic because the number of cases have been increasing since the 60s. There are social changes. These changes include sexual freedom, some changes in moral standards and family unit. Uh, uh, Birth controls were developed, so one of the concerns was pregnancy during sex, and because of the development of these non-barrier contraceptions, people were, their mind was at ease about pregnancy, but what about the diseases? And still, STDs still remain a difficult public health problem. Let's move to slide 48. HPV genital infection. It's the most common sexually transmitted disease in United States. It affects 6 million in United States and 600 million worldwide. Signs and symptoms of the disease. Clinical signs are anogenital warts, warts that we see in the anal area as well as the genital areas. Initially, nearly all of HPV infections are cleared thanks to host cell immune response. And the immune arm, the immune arm of the host cell that clears HPV infection is cell mediated. So we're talking about the phagocytes, T cells, etc. However, most penile infections in men remain subclinical. What does it mean? It means that we do not have signs and symptoms of the disease that alerts the patient to go and seek treatment. And unfortunately, these individuals will serve as carriers to transmit the disease during sex. These subclinical infections may persist and lead to cancer. The variety of cancers in both males and females, including cervix, vulva, penis, and anus. Moving to slide 49. HPV cervical dysplasia and neoplasia. HPV infection initially presents itself as asymptomatic, maybe a little bit of an itching and warts. Weeks or months after sexual contact, the symptoms will develop, and the changes can be detected by a cytologic test, cytocell. So basically, when you think about it, we need to take the cells through a process called pap smear to look for irregular cells. These cytological changes are detected by a type of staining called Papa-Nicolau staining. So we have a swab of cervical smear, we put it in a glass slide, and then we stain it. We look for koilocytotic cells. These are cells with perinuclear cytoplasmic vacuoles. So we see vacuoles in the cytoplasm and around the nucleus of the infected cells. If we see such a cell, they are called koilocytotic. So again, after development of the symptoms, we do a cytologic test, which is we take a cervical smear. So sample is a cervical smear. The procedure is a staining called Papa-Nicolau staining, and we look for koilocytotic cells. Then what do we see? We either see dysplasia, which is 40 to 70% of the cases is mild, and these changes will spontaneously regress. They go away by themselves. Dysplasia changes in the cell, whereas neoplasia are more progressive cellular changes, and these changes will result in cervical interepithelial neoplasia, or CIN, which is cancer of the cervix. And the neoplastic changes will take a little bit longer, 1 to 4 years. Therefore, it is highly recommended for the females to do routine pap smears. Routine pap smears are not only for screening, but more importantly, for prevention, and even more important, for the treatment in the case that there is an underlying disease going on. Moving to slide 50, I have included a little bit of a description of dysplasia and neoplasia. Just take a look at it, and then I also have some information about a benign and a malignant tumor here. Moving to slide 51, HPV cervical carcinoma timeline. It means that how much time does it take from an infection with human papillomavirus to the development of a cervical carcinoma or cancer of the cervix. The disease develops very slowly because there are multiple factors involved. It can be the environment, the chemical composition and the environment of the cervix itself, host cell immunity, etc. And it can cause persistent infection. So we have persistent HPV infection. This infection is where we have viral DNA being integrated into the host cell DNA and is present in the host cell. The most frequently serotypes of HPV that we see in cervical carcinoma are HPV 16 and 18. Peak incidence of the disease is during adolescence and young adult years, or soon after initiation of sexual activity. A subset of infections are precancerous, meaning that there is a time delay after initial HPV infection until the virus causes cancer. And cancer occurs in long intervals. It takes some time to develop, but once it develops, it's very invasive, and it not only progresses very rapidly, but it has high metastatic index, so it can metastasize to other areas of the body. So as you can see in the picture here, the y-axis is incidence, the x-axis is the age or year. As we can see, the peak is during 15, 16, 17, up to 20 years probably, where is soon after initiation of sexual activity. Sometimes we have these instances that we have an infection, but the infection clears by itself. Those cells are dysplastic at this point, but sometimes we have precancerous lesions, that these precancerous lesions will convert into cancer, and it's a very invasive type of cancer. Moving to slide 52, there is a vaccine for HPV called Gardasil. Oh, what's a component of Gardasil? It's a non-infectious recombinant vaccine of capsid L protein. So there is a protein on the capsid of the virus called L1. We take that, through DNA recombinant technology, we make lots of it in the laboratory, which is non-infectious because it's only capsid protein. It does not have DNA. And upon injection, it forms virus-like particles, or VLPs. These VLPs do not have DNA, therefore they're very safe. The vaccine is quadrivalent. It's effective against HPV 6, 11, 16, and 18. These are serotypes. And recently it has been approved for serotype 9 as well, Gardasil 9. So what does it do? It prevents HPV persistent infection and development of genital precancerous lesions. It was initially, the Gardasil was initially recommended only for females, but now it's also recommended for males as a preventive measure before being sexually active. However, because we have something that may work, there is not a green light, and there is no encouragement for early or promiscuous sex. The good news is that it prevents HPV-associated genital burden and cancer. Moving to slide number 53. The question is why we do not have lines for vaccines anymore. Perhaps CVS and Walmart and Rite Aid are competing with each other in providing it to us, which is a very nice and pleasant thing. So I understand that this lecture is being a little bit long, so this would be for 2 sessions. Don't worry about the content. For this week, you will have one more lecture that I will post later on. So let's talk a few words about cellular transformation and cancer. Cancer is uncontrolled growth of a cell, and it makes clones of tumor cells. What does it mean? Clone means that it originates from one single cell. So hypothetically, all of the cells in a tumor mass should be identical. However, this is not the case because we have genetic instability and we have a concept called tumor heterogeneity. Cancer has the potential to metastasize, meaning that it can travel from its original site to other sites of the bodies and involves other tissues and organs. 1 in 3 people will develop cancer in their life, and there are over 200 types of cancers that affect different tissues of the body. A high percentage of the cancers, about 80% of them, are due to carcinogens, those agents that can cause cancer. These include chemicals, tobacco, asbestos, radiation, and so on. And only 20% of the cancers are associated with viruses. These viruses cause, obviously because of their integration into host cell genome, are capable of causing genetic changes, increase the risk of developing cancer, and it is not usual that, but it's correlative, and we have carcinogens, co-carcinogens, meaning that we have virus infection going on, and then we also have additional factors, co-carcinogens, that aid the process of transformation. So how does a DNA virus infection leads to cellular transformation? On the left, on the right, you see a tumor virus DNA that causes infection. Upon infection, it will integrate into host cell DNA, and host cell DNA will undergo transcription. It produces host proteins as well as those genes and proteins encoded by the tumor virus DNA that being integrated into the host DNA, resulting in the translation of those viral proteins. And these viral proteins are tumorigenic or cause cancer. So moving to the left side of the slide, either a part or the whole viral DNA will be integrated into host cell DNA, and viral genes, those genes that are for cellular transformation, will be transcribed into an mRNA. mRNA will be translated into those proteins that have that their main function is to change host cell dynamics and convert a normal host cell into a tumor cell. And loss of growth inhibition is, in some instances, defined as cancer. So when you think about it, what are some of these viral proteins that can be produced, or what are their functions? We will touch on some of these in future slides, but I guess we mentioned that papillomaviruses produce E6 and E7 proteins, and through their function, we get cancer. Moving to slide number 56. Some definitions and terminologies. Oncogenes. If you break down the word onco, cancer, genes that cause cancer. So gene by itself cannot. Genes code for proteins that can cause cancer. We have these both in viruses and their counterparts in the host cell. Those viral oncogenes are called V for virus, onc for oncogene, V-onc, and the cellular counterpart is C-onc. So let's talk about viral oncogenes, or V-oncs. It's a viral gene that is responsible for the oncogenicity of virus, or the ability of virus to cause cancer. V-onc in RNA tumor viruses are altered cellular genes that are acquired from host cell genome, whereas in DNA tumor viruses are not cellular counterparts. And these are gene products, usually functions in viral replication. Moving to slide number 57, I provided a little bit more definition of an oncogene and a proto-oncogene. It won't hurt you guys to read through it. Slide number 58, additional information on proto-oncogenes. I don't want you to go through all the details of it, like Philadelphia chromosome, BCR, ABL. I'm sure you've gone over this in the hematology course. Moving to slide number 59, the lecture summary slide. Polyoma viruses are found in monkeys, such as SV40 virus, and humans, JC and BK virus. Human papillomavirus, HPV, causes genital warts, tumors, or cancers. The oncogene theory was proposed from studies of tumor virus genes that encode cell transformation proteins. What does it mean? We had some genes encoded by the viruses that we saw that the product of those genes, or the proteins being made off of those genes, can cause cancer in the cells. 20% of human cancers are associated with viral etiology, meaning that a virus is the etiology or the cause of the cancer. However, a virus by itself is not sufficient. We need co-carcinogens. We have oncogenes from those viruses that cause cancer, and their genome is RNA or RNA tumor viruses. These viral oncogenes are altered host cell genes that are most likely acquired from host cell genome, whereas the DNA tumor virus oncogenes, or VONCs, a prime example of it would be SV40 large T antigen, is required for replication of the virus itself, and it's not acquired with the host cell genome. Okay, with this, I stop the lecture for today. If you have any questions, please email me. Let me know if you have any questions, and please let me know if this is a good way for us to communicate, or do you want me to have my lectures at 8 o'clock in the morning. Thank you so much for your attention, and I will upload another audio file for this week. Have a pleasant day, stay safe and healthy out there. Take care.